Neanderthal secrets. What their DNA reveals about us. The shadow in the mirror. For the vast majority of human history, we believed we were unique. Homo sapiens, the wise man, stood alone as the pinnacle of evolution, a singular creation or the sole survivor of a linear march from primate to master of the planet. We looked at the archaeological record and saw a clear distinction between our sophisticated tools, our art, our complex societies, and the crude remnants of those who came before. Among those predecessors, none captured the imagination or the derision of the public quite like the Neanderthals. Since their discovery in the 19th century, they were cast as the ultimate others. They were the knuckle-dragging brutes of popular culture, the hunchbacked cavemen who communicated in grunts and lacked the spark of true humanity. They were the losers of the evolutionary game, an evolutionary dead end that perished because they could not compete with the intellect and agility of modern humans. This view persisted for over a century, reinforced by early anatomical reconstructions that were based on arthritic skeletons and a deep-seated anthropogenic arrogance. We wanted them to be different. We needed them to be inferior to justify our own dominance. But science has a way of dismantling our most cherished myths, and the revolution began not with a shovel in the dirt, but with a pipette in a clean room. The decoding of the Neanderthal genome has fundamentally altered our understanding of who they were, and more importantly, who we are. It turns out that the Neanderthals did not just vanish into the mists of prehistory. They are still here. They walk among us, and they live within us, the Valley of the New Man. To understand the genetic revolution, we must first understand the physical discovery that started it all. In 1856, quarry workers in the Neander Valley near Dusseldorf in Germany blasted open a small cave known as the Klein Feldhofer Grot. Inside, they found a collection of bones that they initially dismissed as the remains of a cave bear. A local teacher and amateur naturalist named Johann Karl Fulrat examined them and realized they were human, yet distinctively different. The skull cap was thick with heavy brow ridges. The limb bones were stout and bowed, suggesting immense strength. Fulrat, along with anatomist Hermann Schaffhausen, proposed that these were the remains of an ancient race of humans. The scientific establishment was skeptical. Some argued it was a pathologically deformed modern human, perhaps a Cossack soldier who had died during the Napoleonic Wars, his legs bowed from a lifetime in the saddle, his brow furrowed from pain. It took decades for the scientific community to accept that this was indeed a separate species or subspecies of human. They were named Homo neanderthalensis. As more fossils were uncovered across Europe and Western Asia, a picture emerged of a people adapted to the harsh, glacial environments of the Pleistocene. They had large noses to warm and humidify cold, dry air. They had barrel chests and short limbs to conserve heat. They were built for survival in a world of ice and megafauna. Yet, despite these physical descriptions, their cognitive life remained a mystery, obscured by the prejudice that they were merely dumb beasts. It was the study of their DNA that would eventually give them a voice, the impossible puzzle of ancient molecules. The idea of sequencing the DNA of a creature that died 40,000 years ago was, for a long time, considered science fiction. DNA is a fragile molecule. Upon death, the repair mechanisms of the cell cease to function. Enzymes begin to degrade the genetic material, chopping the long strands of the genome into tiny fragmented pieces. As water, oxygen, and microbial activity further assault the code, chemically modifying the bases and making them difficult to read. After thousands of years, what remains is often less than 1% of the original endogenous DNA, swamped by a vast ocean of contamination from soil bacteria, fungi, and the modern humans who handled the bones. The man who would solve this puzzle was Svante Pääbo, a Swedish geneticist who dedicated his career to the field of paleogenetics. His early work with Egyptian mummies proved that ancient DNA could be extracted, but it also highlighted the massive problem of contamination. A single flake of skin or a droplet of sweat from a modern researcher could overwhelm the ancient signals. Pabo and his team at the Max Planck Institute for Evolutionary Anthropology in Leipzig spent decades refining their techniques. They developed clean rooms that resembled the high-security containment facilities used for studying deadly viruses. They utilized ultraviolet light to destroy surface DNA on fossils. They created complex algorithms to distinguish between the damaged, fragmented ancient DNA and the long, pristine strands of modern contaminants. The breakthrough came when they moved from analyzing mitochondrial DNA, which is abundant but offers limited information, to the nuclear genome, which contains the complete blueprint of an organism. In 2010, after years of painstaking work, Pabo and his colleagues published the first draft sequence of the Neanderthal genome. The results sent shockwaves through the scientific world. The ghost in the machine. The sequencing of the Neanderthal genome allowed for a direct comparison with the genomes of living humans. The expectation among most geneticists, based on the out-of-Africa theory, was that modern humans emerged from Africa roughly 200,000 years ago and spread across the globe, replacing archaic populations like Neanderthals without mixing. This was the replacement model, or the total replacement hypothesis. It posited a clean break. We were a new species, and they were the old. But when the data was analyzed, it told a different story. The researchers found that non-African populations living today share between 1 and 4% of their DNA with Neanderthals. This shared genetic material was not found in sub-Saharan African populations whose ancestors had never left the continent. The implication was undeniable. When modern humans migrated out of Africa and moved into the Middle East and Europe, they encountered Neanderthals. And they did not just fight them or ignore them. They mated with them. This interbreeding event, or admixture, likely occurred in the Levant region roughly 50 to 60,000 years ago, before the diverging human populations spread into Europe, Asia, and the Americas. This means that everyone of non-African descent possesses a genetic legacy from these archaic humans. We are hybrids. The discovery shattered the notion of pure lineages. It suggested that human evolution was not a branching tree with dead ends, but a braided stream, where populations diverged and then merged again. The immune advantage. Once the fact of interbreeding was established, the question shifted to what exactly we inherited. Evolution is a ruthless editor. If a gene does not provide an advantage, or if it is harmful, it is eventually weeded out of the population over generations. The fact that Neanderthal DNA has persisted in our genomes for tens of thousands of years suggests that some of it was beneficial. One of the most significant gifts we received from our Neanderthal cousins relates to our immune system. Neanderthals had been living in Europe and Western Asia for hundreds of thousands of years before modern humans arrived. They had adapted to the local pathogens, the viruses, bacteria, and parasites unique to those environments. Modern humans, emerging from the tropical environment of Africa, would have been immunologically naive to these northern threats. Interbreeding provided a shortcut to immunity. By mating with Neanderthals, modern humans acquired variants of genes in the human leukocyte antigen, or HLA complex. These genes play a critical role in helping the immune system recognize and destroy foreign invaders. Those early humans who carried the Neanderthal immune variants were more likely to survive the unfamiliar diseases of Eurasia and pass those genes on to their children. Today, these archaic genes are still active in our bodies, helping us fight off infections. However, this inheritance is a double-edged sword. The same hyperactive immune response that saved our ancestors from ancient parasites can, in the modern sterile environment turn against the body itself. Studies have linked Neanderthal genetic variants to an increased risk of autoimmune diseases, allergies, and asthma. The sneeze you suppress in ragweed season may be a direct echo of a prehistoric encounter in a limestone cave. Skin and hair, and the cold north. Beyond immunity, Neanderthal DNA played a crucial role in our physical adaptation to the northern latitudes. Africa is a continent of intense sunlight, and the ancestors of modern humans had dark skin to protect against damaging ultraviolet radiation. Europe and Asia, with their lower light levels and colder climates, presented a different challenge. To synthesize enough vitamin D, skin needed to be lighter to absorb more sunlight. Neanderthals had already evolved adaptations for this environment. Genetic analysis has identified Neanderthal alleles involved in the production of keratin, the protein that makes up our skin, hair, and nails. These variants likely helped early modern humans adapt their skin to the colder, drier climates of the north, perhaps by making the skin tougher or improving its ability to regulate moisture and heat. It is also likely that some genes influencing hair color and texture came from our archaic cousins. While the popular image of a Neanderthal is often dark-haired, genetic evidence suggests they had a diversity of pigmentation, including red hair and fair skin, though the specific genetic mutations for red hair in Neanderthals are different from those common in modern humans, an example of convergent evolution. Nevertheless, the genetic toolkit for surviving the Eurasian winter was partly borrowed from those who had called it home for millennia. The metabolic legacy. Our metabolism, the way we process food and store energy, also bears the signature of the Neanderthal. In the harsh, resource-scarce environment of the Pleistocene, the ability to store calories efficiently was a survival necessity. Neanderthal variants associated with lipid catabolism and the storage of fats have been identified in modern populations. In a world where food was intermittent and freezing temperatures demanded high energy expenditure, these genes were lifesavers. They allowed our ancestors to maximize the caloric value of the meat and gathered plants they consumed. However, in the modern world of caloric abundance and sedentary lifestyles, these once beneficial traits have become liabilities. Some of these Neanderthal gene variants are now associated with an increased risk of type 2 diabetes and obesity. The genetic programming that helped a hunter-gatherer survive a glacial winter is now contributing to a global health crisis in the 21st century. This phenomenon highlights the mismatch between our evolutionary heritage and our current environment. We are operating with Stone Age hardware in a digital age world. The mind of the other. Perhaps the most tantalizing question concerns the Neanderthal mind. Were they cognitively different from us? Did they think, feel, and dream as we do? For decades, the archaeological consensus was that Neanderthals lacked the capacity for symbolic thought. They were seen as having a static culture, using the same stone tools for hundreds of thousands of years without innovation. They were contrasted with the explosion of art and advanced weaponry associated with the arrival of modern humans in the Upper Paleolithic Revolution. However, genetic and new archaeological evidence is challenging this view. The FOXP2 gene is critical for language and speech development in humans. When researchers looked at the Neanderthal genome, they found that they shared the exact same two mutations in the FOXP2 gene that are unique to modern humans. This suggests that, from a genetic standpoint, they had the motor machinery for complex speech. Whether they had the neural wiring to support complex grammar is still debated, but the hardware was there. Furthermore, recent archaeological finds have shown that Neanderthals made jewelry from eagle talons, used pigments like red ochre, and perhaps even created cave art. In caves in Spain, abstract paintings have been dated to long before modern humans arrived in Europe, implying Neanderthal authorship. These behaviors require symbolic thinking, the ability to let one thing represent another, which is the bedrock of human culture. While there are regions of the modern human genome that are notably devoid of Neanderthal DNA, specifically those areas associated with the brain and cognitive development, this does not necessarily mean Neanderthals were unintelligent. It suggests that there were specific cognitive differences, perhaps in social connectivity or creativity, that gave modern humans a slight edge. But the gap is narrowing. They were not mindless robots. They were a different kind of human, with their own culture, their own rituals, and their own complexity. The dark side of inheritance. Not all the secrets revealed by Neanderthal DNA are benign or purely historical. As we delve deeper into the medical implications of our archaic heritage, we find associations with a wide array of modern conditions. One of the most striking discoveries came during the COVID-19 pandemic. Researchers identified a gene cluster on chromosome 3 that is a major risk factor for severe respiratory failure after infection with the SARS-CoV-2 virus. This risk variant is inherited from Neanderthals. Individuals carrying this genetic segment were significantly more likely to require hospitalization and intubation. It is a tragic irony that a genetic legacy which likely helped our ancestors fight off ancient RNA viruses has proved detrimental against a novel modern virus. This finding underscores the randomness of evolution. A trait is only good or bad in the context of a specific environment. Beyond infectious disease, Neanderthal DNA has been linked to the risk of blood clots, which would have been an advantage for healing wounds quickly in a dangerous prehistoric world, but is a risk factor for stroke and heart attack today. There are also associations with neurological and psychiatric conditions, including depression and addiction. The influence of these ancient genes is subtle, often affecting the regulation of other genes rather than coding for proteins directly, but their cumulative impact on human health is significant. It forces us to view our medical history through an evolutionary lens. We are not a perfect design. We are a patchwork of adaptations, some of which have outlived their usefulness. The Denisovan connection. The story of Neanderthal DNA is inextricably linked to another major discovery that occurred in the same Siberian cave that gave us the first high-quality Neanderthal genome. In Denisova Cave, archaeologists found a tiny fragment of a finger bone belonging to a young girl. When Svante Pabo's team sequenced the DNA from this bone, they expected it to be Neanderthal or modern human. Instead, it was something else entirely. The DNA sequences diverged from Neanderthals and modern humans roughly 400,000 years ago. They had found a new species of human, identified solely by its genetic code. They were named the Denisovans. Further analysis revealed that Denisovans were a sister group to the Neanderthals, splitting from them after their common ancestor left Africa. While Neanderthals spread west into Europe and the Middle East, Denisovans spread east into Asia. And just as modern humans interbred with Neanderthals, they also interbred with Denisovans. Today, traces of Denisovan DNA are found in populations across Asia, Melanesia, and among indigenous Australians. In some populations in Papua New Guinea, the Denisovan contribution is as high as 5%. This discovery expanded the cast of characters in the human story. It revealed a Eurasia populated by multiple distinct groups of archaic humans who interacted and mated. The Denisovans also bequeathed genetic gifts. A variant of the EPAS1 gene, found in Tibetans, allows them to live at high altitudes with low oxygen without suffering from the thick blood that plagues lowlanders. This gene was inherited from Denisovans. It is a striking example of how acquiring genetic material from locally adapted archaic populations facilitated the global expansion of modern humans. The purging of the genome. While we focus on the DNA that remains, it is equally instructive to look at the DNA that is missing. When we map Neanderthal sequences onto the human genome, we see a landscape of peaks and valleys. The peaks are the regions where Neanderthal DNA has been retained because it offered an advantage or was neutral. The valleys, or deserts, are large stretches of the genome where Neanderthal DNA is completely absent. These deserts are particularly prominent on the X chromosome and in genes expressed in the testes. This pattern is a hallmark of a phenomenon known as hybrid sterility. When two distinct species or widely divergent populations mate, male offspring are often sterile or have reduced fertility. This biological barrier suggests that by the time modern humans and Neanderthals met, they were on the brink of becoming biologically incompatible. They were at the very edge of the species boundary. Natural selection worked aggressively to purge these fertility-reducing genes from the human population. Over thousands of years, the bad Neanderthal DNA was washed away, leaving only the compatible or beneficial segments. This process of purifying selection tells us that while we could mate, it was not a seamless biological merger. It was a chaotic, biologically costly process that only succeeded because the advantages of the good genes outweighed the reproductive costs of the bad ones. This gives us a precise definition of our biological distance from them. We were cousins, but distant enough that our reunion was fraught with biological difficulty. The social structure of the ancients, genetic sequencing, has also allowed us to peer into the social lives of Neanderthals in ways that archaeology alone never could. In 2022, researchers published the genomes of 13 Neanderthals found in 2 caves in the Altai Mountains of Siberia, Chagirskaya and Okladnikov. This was the first time researchers were able to analyze a community of Neanderthals who lived at the same time. Among the remains, they found a father and his teenage daughter, as well as other close relatives. The snapshot of a Neanderthal family offered profound insights. The genetic diversity of the Y chromosomes, passed down from fathers, was very low, while the mitochondrial DNA passed down from mothers was much more diverse. This pattern strongly suggests a patrilocal social structure. In this system, men stayed in the groups they were born into, while women moved between groups to find mates. This is a common pattern in many human hunter-gatherer societies, and even in chimpanzees and bonobos. It paints a picture of small, interconnected bands of Neanderthals, with women serving as the genetic bridges between distinct communities. It humanizes them. We can imagine the young women leaving their families to join new groups, carrying with them not just their genes, but knowledge, stories, and traditions. It refutes the idea of Neanderthals as isolated, solitary wanderers. They had networks, they had families, they had societal rules. The twilight of a species. If Neanderthals were so well adapted, intelligent, and capable of interbreeding with us, why did they disappear? This remains one of the great debates of paleoanthropology. The genetic evidence helps us refine the theories. We know that the Neanderthal population was never very large. Estimates suggest that at any given time, there were perhaps only 10,000 to 70,000 Neanderthals spread across the vast expanse of Eurasia. They lived in small, isolated populations with low genetic diversity, making them vulnerable to inbreeding and stochastic events. When modern humans arrived, they brought with them not only higher population densities, but also distinct cultural adaptations, such as projectile weaponry and perhaps more complex social networks that allowed for trade and resource buffering. The extinction of the Neanderthals was likely not a single violent event, a prehistoric genocide. It was more likely a slow process of assimilation and competitive exclusion. As modern humans expanded, Neanderthal populations were absorbed. Their distinct lineage was diluted generation by generation until it persisted only as a ghost sequence within our own DNA. We did not necessarily kill them all. In a very real sense, We became them, and they became us. The pure Neanderthal disappeared, but the hybrid survived. Climate change also played a role. The period around 40,000 years ago saw rapid and severe fluctuations in climate. The open steppe tundra environments that Neanderthals hunted were changing. Modern humans, with their more diverse diet and flexible tools, may have simply been better at adapting to this instability. It was a death by 1,000 cuts. Demographic swamping, assimilation, climate stress, and competition. The philosophical mirror. The realization that we are part Neanderthal has profound philosophical implications. It challenges the concept of human purity. For centuries, racists and eugenicists obsessed over bloodlines and purity, often using the Neanderthal as a foil for the superior modern human. Science has turned this on its head. It is the Europeans and Asians, often the proponents of such Eurocentric superiority, who carry the DNA of the caveman. Meanwhile, sub-Saharan African populations, often historically marginalized by Western science, represent the lineage of Homo sapiens that did not mix with these northern archaics. This genetic reality dismantles the biological basis of racial hierarchies. It reveals a humanity that is deeply interconnected, a product of migration and mixture. It also forces us to reconsider our definition of human. If Neanderthals could speak, make art, care for their sick, as evidenced by healed fractures in fossils, and produce fertile offspring with us, were they truly a different species? Or were they just a different kind of people? The line between us and them has blurred to the point of irrelevance. We are part of a continuum. The secrets in our DNA tell a story of unity, not division. We are the result of a complex history of interaction, where different groups of hominids met, fought, loved, and merged. The future of the past. The field of paleogenetics is moving at a breakneck speed. We are no longer limited to bones. Scientists can now extract ancient DNA from cave dirt, identifying which species occupied a site without ever finding a fossil. This sediment DNA is opening up vast new chapters of prehistory, allowing us to map the presence of Neanderthals and Denisovans across time and space with unprecedented resolution. We're also beginning to study the epigenetic markers on ancient DNA, the chemical switches that turn genes on and off. This could allow us to reconstruct not just the anatomy of Neanderthals, but how their bodies actually functioned at a cellular level. There is even talk, mostly theoretical and ethically fraught, of using CRISPR technology to reintroduce Neanderthal variants into human cells in the lab to study their effects more directly. We are creating Neanderthalized organoids, mini brains grown in dishes, to see how their neurons develop differently from ours. This research holds the promise of unlocking the secrets of our own brain evolution, identifying the specific genetic changes that gave us our unique cognitive capacities. But it also raises ethical questions about the dignity of our ancestors and the risks of resurrecting the biology of the past. The final legacy. In the end, the secrets of the Neanderthals are not locked away in dusty museum cabinets. They are flowing through the veins of billions of people alive today. When you fight off a flu virus, when your skin burns or tans in the sun, when you struggle with a mood disorder or an autoimmune disease, you are experiencing the echo of a life lived 40,000 years ago. The Neanderthals were survivors. They endured ice ages and predators for hundreds of thousands of years. They were masters of their world. And in a way, they succeeded in the ultimate evolutionary goal. They passed their genes on. They are not extinct. They are repurposed. We are the vessel for their legacy. The study of their DNA has humbled us, stripping away our pretensions of uniqueness and revealing the deep, tangled roots of the human family tree. It teaches us that to be human is to be a hybrid, a mosaic of all those who came before. As we look to the future, carrying their code within us, we are a testament to their resilience. The shadow in the cave was not a monster. It was a grandfather, a grandmother, a part of ourselves waiting to be discovered. The biological mechanism of introgression. To fully grasp the magnitude of the Neanderthal contribution, we must delve into the mechanics of introgression. This term describes the movement of a gene flow, from one species into the gene pool of another by the repeated backcrossing of an interspecific hybrid with one of its parent species. In the case of humans and Neanderthals, the initial hybridization events were likely rare. We are not talking about rampant, widespread merging of populations, but rather distinct pulse events. Genetic modeling suggests that the primary introgression event occurred roughly 50,000 to 55,000 years ago. However, recent data implies there may have been multiple pulses. For example, the ancestors of East Asians may have encountered Neanderthals a second time after they separated from the ancestors of Europeans, which would explain why East Asian populations today have slightly more Neanderthal DNA, roughly 20% more than European populations. This differential is not due to selection alone, but points to a complex history of migration and interaction. The initial hybrids would have been half and half. But if those hybrids stayed within the modern human population and mated with modern humans, the percentage of Neanderthal DNA would halve with each generation. 50%, then 25, then 12.5. Within a few centuries, the contribution would dilute to the low percentages we see today. The fact that it stabilized around 2% implies that after the initial purging of deleterious genes, the remaining segments were either neutral or advantageous enough to be maintained. This maintenance is not passive. It is an active retention of genetic material that proved useful. The structural variants and the hidden code. Early analysis focused on single nucleotide polymorphisms, SNPs, which are changes in a single letter of the genetic code. However, new sequencing technologies allow us to look at structural variants, insertions, deletions, and larger rearrangements of DNA. These structural changes can have a much more profound impact on gene function than single-letter swaps. It turns out that Neanderthals contributed a significant number of these structural variants to the human genome. Some of these variants affect the regulation of gene expression, the volume knobs that determine how much of a protein is made. This is a critical distinction. We are not just talking about different proteins, but different ways of using the proteins we have. For example, Neanderthal regulatory elements have been found to influence the expression of genes involved in the nervous system and the development of the skull. This suggests that while the coding regions of our brain genes might be purely human, the control mechanisms might have a Neanderthal influence. This adds a layer of complexity to the Neanderthal mind debate. It is not just about the genes you have, but how you use them. The investigation of these regulatory regions is the new frontier in paleogenomics. It is akin to finding the conductor's notes on a musical score, explaining how the orchestra of our biology should play. The landscape of the X chromosome. One of the most telling features of Neanderthal ancestry is its uneven distribution across the chromosomes. As mentioned, the X chromosome is remarkably poor in Neanderthal ancestry compared to the autosomes, non-sex chromosomes. In many species, the X chromosome is a hotspot for genes related to male reproduction. The scarcity of Neanderthal DNA here is a classic signal of hybrid incompatibility. It suggests that male hybrids were likely sterile or had very low fertility. This follows Haldane's rule, which states that in the offspring of two different animal races, if one sex is absent, rare, or sterile, that sex is the heterogametic sex. In mammals, the male XY. This implies that the flow of Neanderthal genes into the modern human population was primarily driven by female hybrids. The Neanderthal men who mated with human women may have had sons who could not procreate, ending that lineage. But their daughters could. This creates a maternal bias in the introgression story. It forces us to consider the gender dynamics of these ancient encounters. Were they consensual? Were they part of exchange rituals? Or were they the result of conflict? While the DNA cannot definitively answer questions of consent, the biological filter of the X chromosome tells us which lineages survive to become part of us. The chronobiology of the past. Another fascinating area of research involves chronobiology, our internal body clocks. Neanderthals living in high latitudes experienced extreme variation in daylight hours, from the long days of summer to the long nights of winter. Modern humans from Africa were adapted to a 12-hour day/light cycle that remained relatively constant. Adaptation to the northern light cycle involves genes that regulate the circadian rhythm. Research has shown that Neanderthal alleles are enriched in genes that control our body clock. These variants are associated with being a morning person or an evening person. The tendency to wake up early may be an adaptation inherited from Neanderthals. To maximize daylight activity in the short winter days of Pleistocene Europe. This alignment of our internal clocks to the Eurasian latitude is a direct functional legacy. It affects our sleep patterns, our alertness, and potentially our susceptibility to seasonal affective disorder. The way we experience time and sleep is partly dictated by the survival strategies of our archaic ancestors. The enduring mystery of the superarchaic. The sequencing of the Denisovan and Neanderthal genomes revealed yet another twist. When geneticists analyzed the Denisovan genome, they found traces of DNA from an even older, unknown human species, a superarchaic hominid. Denisovans had interbred with this ghost population, which had branched off from the human tree over 1 million years ago. Some of this superarchaic DNA might have been passed from Denisovans to modern humans in Asia. This implies a deep, nested Russian doll of inheritance. We contain Neanderthal DNA, which contains traces of older interactions. The identity of this superarchaic human is a mystery. It might be Homo erectus, or another species yet to be identified. This finding reinforces the idea that the Pleistocene world was a melting pot of hominid forms. It was not a lonely planet. It was crowded with different experiments in being human, all interacting and exchanging genetic material. The deeper we look, the more complex the web becomes. The toolmaker's hands. We often focus on the brain, but the hands are equally important. The dexterity required to make stone tools, to knap flint into razor-sharp flakes, requires specific anatomical traits. Neanderthals had hands that were distinct from ours, broader fingertips, and a thumb with a different range of motion. They were capable of a power-precision grip. While they might not have had the delicate needle-threading capability of modern humans, their hands were incredibly strong and capable of fine motor control, suited to their technology. Genetic variants affecting bone density and muscle attachment in the hands have been traced, but the signal is complex. What is clear is that the tools they made were not primitive. The level law technique, a method of preparing the stone core to strike off a specific shape of flake, requires abstract planning and visualization. You have to see the tool in the stone before you strike. This cognitive motor loop is a shared heritage. While we replaced their tool industries with our own, the underlying capacity for technology, the interface between hand and mind, is something we share. The discovery of birch tar pitch used by Neanderthals to haft spear points is particularly revealing. Making birch tar requires a controlled dry distillation process at roughly 350 degrees Celsius. It is the first known industrial process. It implies a deep understanding of materials and fire, We can no longer dismiss them as technologically incompetent. The echo of the voice. Language leaves no fossils, but it leaves genetic clues. Beyond FOXP2, researchers are looking at other genes involved in vocal tract formation and auditory processing. The Neanderthal ear bones, ossicles, are shaped slightly differently than ours, but modeling suggests their hearing was tuned to the same frequencies as human speech. They were not deaf to the sounds we make. The physical capacity to produce vowels and consonants was likely there, perhaps with a slightly different timbre due to the shape of the skull and nasal cavity. The real question is the software. Did they use syntax? Did they tell stories? The presence of the human version of FOXP2 is a strong argument for complex communication. It is unlikely that such a metabolically expensive biological machinery would be maintained if it wasn't being used. If they spoke and we spoke, did we learn from them? Are there loan words from Neanderthal languages hidden in the proto-languages of Eurasia? This is pure speculation, as languages evolve too fast to preserve traces over 40,000 years. But the possibility of cultural exchange, facilitated by language, is a tantalizing thought. The medical frontier, pharmacogenomics. In the realm of precision medicine, Neanderthal DNA is becoming a clinically relevant variable. Pharmacogenomics is the study of how genes affecting a person's response to drugs. Variants in the cytochrome P450 family of enzymes, which metabolize many common medications, differ between populations. Some of these variants have archaic origins. Knowing a patient's Neanderthal carrier status could theoretically influence the dosage of painkillers, blood thinners, or antidepressants. For instance, a specific Neanderthal variant might make a person metabolize a drug too quickly, rendering it ineffective, or too slowly, leading to toxicity. As we move toward a future where medical treatment is tailored to the individual genome, the Neanderthal panel might become a standard part of a checkup. We are treating the patient not just as a modern human, but as a biological composite, the burden of the skin. Our largest organ, the skin, is a primary interface with the world. We discussed keratin, but there is more. Neanderthal variants are linked to the risk of developing keratosis, distinct precancerous lesions. They are also linked to corns and calluses. This seems trivial, but it points to the specific adaptations of Neanderthal skin to physical abrasion and cold. Their skin was likely thicker and more robust. In our comfortable shoe-wearing lives, these adaptations manifest as dermatological annoyances. Furthermore, the genetics of pigmentation is complex. While we mention they had variable skin tones, the specific alleles for tanning ability, the skin's reaction to UV radiation, also show archaic influence. The ability to tan rather than burn is a crucial adaptation for populations moving between seasons of low and high UV intensity. We owe some of our sun smarts to them. The psychological dimension. Is our personality influenced by our ancient cousins? This is the most controversial and difficult area of study. Some studies have found correlations between Neanderthal variants and behavioral traits, like smoking status, enthusiasm, and even loneliness. However, these associations are weak and often confounded by other factors. It is dangerous to fall into biological determinism. Being 2% Neanderthal does not make you aggressive or solitary. However, the fact that genes expressed in the brain, genes involved in neurotransmitter regulation, are among the introgressed regions suggests that the architecture of our mood and behavior has archaic pillars. The depression link mentioned earlier is likely tied to inflammation. The immune system and the brain are deeply connected. Inflammation can drive depressive symptoms. If Neanderthal immunity genes make us more prone to inflammation, they indirectly influence our mental state. It is a cascade effect, from a gene meant to fight a bacterium to a mood state in a modern human. The conservation of the genome. Why do we have so little Neanderthal DNA left? If it was 50% in the first generation, why did it drop to 2%? Why not 10 or 20? This brings us back to the concept of genetic load. The Neanderthal population was small and inbred for a long time before we met them. This accumulation of harmful mutations meant that, on average, a Neanderthal was less genetically fit than a modern human. When the two populations mixed, natural selection acted efficiently to remove the deleterious Neanderthal alleles. The 2% we retained is the cream of the crop, the variants that were either neutral or beneficial. The rest was purged. This massive genetic filtering event is a testament to the power of evolution. It took the best of the archaic genome and discarded the rest. We are the optimized result of this grand experiment, the global perspective. It is important to remember that the Neanderthal legacy is not universal. Sub-Saharan African populations who have the most genetic diversity on the planet have little to no Neanderthal DNA, though recent studies show very small amounts due to back migration of Eurasians into Africa. This distinction was once used to divide humanity, but in the light of genetics, it highlights the incredible diversity of the human story. The purest Homo sapiens are in Africa. The rest of the world are modified variants admixed with archaic groups. This flips the script on the colonial narrative. The standard human genome is the African one. The European and Asian genomes are the ones with the alien insertions. This perspective shift is essential for a scientifically accurate and socially just understanding of human variation. The final synthesis. We are standing at the threshold of a new era in anthropology. The bones are speaking to us in the language of molecules. The secrets of the Neanderthals, their red hair, their immunity, their mourning habits, their susceptibility to disease, their family structures, are now open books. We have moved from digging for stones to mining data. The image of the Neanderthal has been rehabilitated. They are no longer the brutish failures of history. They are our ancestors, our benefactors, and our mirror. They remind us that we are part of nature, subject to the same biological laws as any other creature. They remind us that our dominance was not inevitable. It was a roll of the dice, a combination of luck, migration, and a little bit of help from the cousins we met along the way. As we face the challenges of the future, climate change, pandemics, genetic engineering, we would do well to remember the lessons of the past. We survived because we mixed. We survived because we were adaptable. We survived because we were not alone. The Neanderthals live on, not in the dark caves of the past, but in the bustling streets of the modern world, hidden in the very cells of the people who study them. They are the ghost in the machine, the whisper in the blood, the secret that reveals us to ourselves. The road ahead. The work is far from over. Thousands of ancient genomes are waiting to be sequenced. We will likely find more ghost populations, more intricate patterns of mixing. We will refine our understanding of exactly what every single Neanderthal gene does in the human body. We may even find Neanderthal DNA in places we never expected. The story of human evolution is being rewritten. Not in ink, but in nucleotides. And the title of that story is not The Ascent of Man, but The Weaving of the Human Tapestry. In this tapestry, the Neanderthal thread is thick and strong, adding color and texture to the fabric of who we are. We are the keepers of their secrets, and in understanding them, we understand the profound complexity of being human.